Welcome to Study Loop.
Okay, so 46 wants us to represent in a graph. So I'll put the graph in the corner here as a function of time. Okay, the sort of amount of information, so let's abbreviate that to info, that's sort of being exchanged, okay, like trends. And so I have here on the left already a signal transduction pathway of the epidermal growth factor, right? So part I here, the extracellular first messenger. That's out here, the extracellular first messenger. So one of these growth factors sort of just being out in the atmosphere, and so there's not really much information exchanged right now. It's just sort of existing outside of the cell, right? So if you see here, this double line is our membrane, and so initially at part I, there's not much going on. It's going to be kind of low. Okay, so maybe I is here. And then moving on, when the receptor binds, so then part two, or right, part two here, when binding occurs, well, that causes these conformational changes, right? That sort of the creating a dimer and these intracellular responses. And so there's a bit more going on. So we see an increase in information. Okay, so part two of the increase. Perhaps I can do that in green so we can keep track here. And then the next step are the second messengers. And if you look at them, there's quite a lot going on here, right? So there's this big transmission to these varying second messengers, this phosphorylation, this whole like signal pathway, right? It's pretty involved. It goes from the like MAPK kinase to MAPK kinase to just the MAPK. And so in here we're seeing a large exchange of information. There's quite a lot going on. Okay, so here we're going to see more increase, but that'll be good. And then the cellular response. So here the cell response for this one can be this phosphorylation of targets promoting, this would promote growth. Okay. Also, we have down below here this transcription of gene promoting cell growth and division, right? So we have some stuff going on down there. And so that's also quite a bit of a exchange of information, right? And so we're getting a bit more. And then as we finish out, so this would be our 4. And as we finish here at the end, then the signal halts and degrades, right? So now we're kind of ending things. And so I drew the 5 here above the tick, and I hope that's not too confusing just because I had time written in the way. Okay, so we get this sort of nice curve, and then to annotate it with specific system. Well, since I have this EGF system, it makes sense to me to annotate it that way, and so it's not too hard now that I've sort of color coded it, right? But so then here we're going to have the extracellular first messenger. So this is the epidermal growth factor, so the EGF. Okay, and then once it binds, we get an increase in exchange, right? And then our second messengers, and you can annotate it as involved as you want. You can talk about these transitions. All these different steps in the second messengers are, I'm just going to kind of be brief, right? So it's just the second messengers and that whole pathway that's occurring. Okay, and then And then we get to some cellular responses. So there's still some more information being exchanged. And then after that, the exchange of information sort of decreases. It's not happening anymore. So this is the halting and degrading. So halt. Okay, so hopefully that sort of makes sense when you have a pathway as an example to kind of follow along with here.